Welcome, everybody. Um, my name is Shiva. I'm a postdoc in the University of Texas at Austin, and I'm going to present my research, wherein I try to monitor or track rodent behavior, especially mouse, mouse's behavior, as well as physiology, using ultrasonic guided waves, which are generated by the mouse. So before I actually go into my research as such, I would like to present some of the research which people can very quickly relate, which is similar yet not so similar to my research. So this presentation of the other research I'm going to give is mainly so that it is very easy to relate some of the concepts which will explain my research. So the first is this is one particular type of research where people try wherein when humans walk, like when we walk on a floor of a building, building, we generate waves, really low frequency waves in the floors, and this is picked up using this really low frequency sensors to measure these vibrations, to actually identify the person or monitor the number of occupants in a room or even the gait of that particular person. And here you see this setup, which is on the extreme right, where they show the geophone, which is a sensor used to measure typically seismic waves. So that is used on the floor to measure these vibrations. So when a person walks, this vibration is picked up. And similarly, um, another similar research is when we lie down and sleep on a cot or somewhere, our breathing as well as heartbeats again induce vibrations in the bed frames. And here, similarly, they use this geophones, very low frequency sensors, to detect the heart rate as well as the breathing rate. So, so the idea is that is like when humans, uh, uh, do anything like humans' physiology as well as behavior generate this really low frequency vibrations, which can be picked by any of these sensors to monitor a lot of host of these parameters. Similarly, when it comes to rodent behavior, a few years ago, some of the researchers tried to pick this really low frequency vibrations. So what they did is, in the schematic of their experimental setup is shown here, is they have this floors where the mice stands or sleeps, coated with PVDF films, which actually allows you to measure the low frequency vibrations. And with this one, they were able to, first of all, track the heart, the breathing rate when track the low frequency vibration when the mice was actually sleeping. So they anesthetized the mice and then they mouse and then they tried to measure these vibrations. So you see the nice cyclic signal here. And similarly, when the mice is little awake but not moving, you get this still cyclic, still periodic, but not as periodic as before, these signals. But when it starts moving, you just get a little haphazard but low frequency signal. So this is sort of like what I am trying to do as well. Not exactly this one, but I try to study something a little different, um, uh, 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 a lot more things, not just sleep or something, but behavior, physiology, even track the psychological states using the ultrasonic guided waves. So whatever you saw a little while ago, uh, so you can already see with that low frequency vibration, you have a limit, what you can and cannot measure. So basically, you can measure something which is cyclic, and when the animal is not moving. But with what I'm presenting now is like with my approach, with the guided waves, the information in the guided waves, uh, I would be able to actually measure physiological parameters of a free moving mouse. Also, identify behaviors, classify behaviors and identify hidden variations within behaviors, which even a advanced researcher cannot differentiate visually, as well as track the psychological state of the animal. So when I say that this technology has the potential to do all this stuff, then the question would be like, what is this technology and how can it do all these things? So That is what first I'm going to answer. A little bit of the background on the technology, what these guided waves are, where, how does the mice generate these things, where they come from, how do I measure it, and what properties of these guided waves allow you to track all this really very subtle things about the physiology as well as behavior. So first of all, let us, like I'm going to give a scenario. Suppose say you have this very thin aluminum plate. So the aluminum plate shown in the image is 40 centimeter by 40 centimeter, and its the thickness is 1/8 of an inch. So when the mice, suppose say walks on it, or like if you just put the mice and it applies any kind of force, the way in which this force, the way in which this energy is transmitted in the plate is through guided waves. So in other words, uh, like if there is any force or any kind of a impact on this on a bounded structure, the way in which those energies propagate is through guided waves, ultrasonic guided waves. So an analogy could be like suppose you have the mouse on this plate, and if that guy is say doing something, behavior is engaging in a behavior or walking, and the waves start to propagate. So an analogy would be to think of like a lake where a very calm lake, and then if somebody throws a stone, then wave starts to propagate. Correct. So that is the same thing in when it comes to guided waves. So basically, the way in which energy starts to propagate in a bounded medium. So there is some impact, and then this impact generates waves and they travel in this, and these are guided by the surfaces of the plate. So mainly these waves are actually how their properties are guided by the bounded medium. Here bounded, when I say bound media, I'm talking about the plate has a boundary like upper surface and lower surface. So that is basically the point of guided waves. And we look into the ultrasonic domain because that's where the really fun stuff, like the beautiful properties of these waves lie in. So which I'm going to talk a little while in a little while. So uh, when suppose say like when the mouse walks or engages in behavior, it generates these guided waves, and these guided waves are of two types. One is called the lamp waves, what is seen on the left, and the other is the shear waves, what is seen on the extreme right. So in lamp waves, you basically what you see here is a schematic of the undulations. So in lamp waves, you mostly have part. So this is like a depiction of the thickness of the cross section of the thickness of the plate. And what you have in this lamb wave are these undulations. You have symmetric undulations as well as asymmetric undulations. So when an animal, when the animal walks on the or does anything on the plate, it is going to generate these lamb modes, both symmetric as well as unsymmetric. And then you also it will also sometimes generate the shear modes. So in shear modes, what happens is, unlike the lamb waves, you don't have out of plane vibrations, but you have in plane rotations. So the point is like when animal engages something, it generates this plethora of different types of modes. And and the other interesting thing about these modes is, so here you can see these modes are actually multimodal. That is, suppose say our mouse tries to is on the plate and it rears, and that rearing causes waves to propagate at 500 kilohertz. So at 500 kilohertz, what happens is there will be two modes that will be propagating: an anti-symmetric zero mode and a symmetric zero mode. And similarly, suppose say the mouse is sniffing, and that waves are at 1000 kilohertz. So at that 1000 kilohertz, you'll have four modes. You will have the anti-symmetric zero A0, symmetric S0, A1, S1. So what this multimodal property allows you to do is for one behavior, you have a multi-dimensional data. You have four modes which are generated for one particular behavior. So you can describe the behavior in terms of a multi-dimensional four wave packets. And the other property of this guided waves is it's dispersive. That is, you can see here the velocity of the modes change with frequency. So the point is, like suppose say the rearing happens at 500 kilohertz, and then there is a small sudden a small variation in the way in which animal rears, and if that variation is like at 560 kilohertz, and what you see is the velocity is different. and then the velocity of the wave now will be in different, and also the signature of consequently the signature of the waves will change. So we will be able to identify all these finer details, even variations within a behavior, using these multimodal and dispersive properties. And how does it help us? Like I just explained, you'll be able to point out all the smaller finer details in the behavior. And also, because these signatures because of these properties, the signatures of the voluntary and involuntary action will be different. That is, the waves which are generated from involuntary forces, suppose say like heartbeat or respiration, they will have a certain signature which will be uniquely different from the ones from behavior, and we will be able to differentiate it based on this change in the signatures. So that will enable us to not only differentiate this physiology from behavior, but also track behavior and variations within behavior. So this is the background. And how do we detect this guided waves? So we have this really high sensitive sensors, ultrasonic sensors, which can be bonded to the plates, and that will actually allow you to capture these ultrasonic waves, which are generated by the mice, by the mouse movements of the mouse. And each sensor has a particular frequency range, and they are designed for a particular either measuring lamb modes or the shear horizontal modes. And in the experiment, what we do is we attach these sensors to the bottom of the plate, so that when the mouse moves on the top of the plate, you get a real time immediate monitoring of the waves. So it's sort of like listening to the guided waves generated by the animal. And the other point I want to emphasize is what suppose in this schematic shown here, the mice moves around the open field and the play on the plate, and the sensors detect this continuously, keep detecting this wave packets. So say that there are three wave packets arising from three behaviors. What you don't want is, like, you do a 10-minute open field study, and you don't want to have like a thousands of wave packets in one big signal. You don't want a continuous data where somebody has to process it. Instead, what can be done is, you have a data acquisition system which can actually get each of this wave packet, separate each of this wave packet according to the behaviors. So how it does is, suppose say like we have two wave packets which are generated from two different behaviors. So we tell the acquisition system, we define a threshold. Say like we say like when once um, uh, the amplitude of the generated wave crosses this threshold, acquire it for a certain number of duration, and acquire it for that duration of that waveform, of that wave packet, and store it as a discrete quantity. So basically, what it does is, it takes this continuous waveform, and then and discretizes each of this wave packet into a separate quantity, like a discrete quantity, which is called an AE hit, which is called an acoustic emission hit. So basically, what we do is, we treat the animal like an acoustic source, and each of the generated ultrasonic waves, or each of the generated wave packets, are stored as a discrete hit, so that each wave hit corresponds to a behavior, or a motion, or a involuntary force, like from a physiological process. And what this allows you to do is, instead of we looking at waveforms, the system actually gives you certain features of the waveform. So you don't want to keep looking at all these waveforms. So instead, what it does is, it gives you it extracts some feature, for example the duration of the waveform, or the amplitude of the waveform, or the energy in the waveform. So, and so at the end of the experiment, when you do an open field test, all you have is this discrete quantities for each hit. So when you have these kind of quantities, discrete quantities, you'd be actually able to feed them in a neural network, and then classify the behaviors and such. So that is like sort of the other um, say like another another improvement when it comes to data processing that we are doing. So now here is the actual system, where you see the open field with the aluminum plate, where and the aluminum plate where the sensors are bonded to its bottom surface. and then are connected to the acquisition system, where you will have a real-time monitoring. As the animal moves around, you will see the waveforms, and then the system will automatically calculate the features and then give you the information at the end of the test. So before I go into the results, one small thing I would like to talk is about the sensitivity of how sensitive are these sensors. Like I'm talking about measuring heart rate and tracking behavioral changes in a subtle fashion. How sensitive are these sensors? Can they actually measure the smaller things? So the answer is yes. So typically, these sensors we have used to measure even like small chemical reactions and molecular changes, like study the effects of them. And an example is what we did in our previous study is corrosion monitoring of pipes. So where here you see is like there's a small saltwater tank where we initiate corrosion in this pipe and try to study the loss of thickness and all that. So the test is done over 150 hours, the corrosion test. And what happens is even as the corrosion, there's an onset of corrosion, you will start seeing this acoustic waves which are generated in the structure, and these sensors are able to pick them up. So they are that sensitive. And as other similar studies include, like people study this corrosion in concrete, like there are rebars in the concrete where they get corroded, and people are able to track that. And the other studies are just to show the sensitivity of the sensor is the degradation of silicon electrodes in lithium ion batteries and all that. So the point is like it can actually track chemical changes, like small chemical changes. So tracking behavioral things are, it's not a very big difficulty for these sensors. So now I'm going to just to showcase in experimental run which we did with this in this setup, in this modified setup. So here, in this video, I'm just going to run this for a couple of seconds. So where you can see the mice in the open field, where the base of the open field is the aluminum plate, which is equipped with these sensors. So the mouse you are seeing is a C57. Here. So now you can see that this is the mouse which is exploring the open field. That different behaviors it is exploring, and then there is a rearing, supported rearing, so on and so forth. So all the while so now there is grooming. So all the while the mouse is doing all these things, the waves are being recorded. So now I'm going to show you the results. So the first thing is so in this test, in this particular test, we used something called the R6A sensors. So which has like it's not the complete ultrasonic range, it has only 30 to 100 kilohertz, and this measures the lamb wave. And then we have the SH sensors, which measures the shear wave, shear horizontal wave, and that is rated for 70 to 230 kilohertz. So we are not measuring the entire spectrum, but a limited spectrum. And we used a threshold of 35 dB. So basically, we say to the system, start acquisition after once the signal crosses 35 dB. So if there are signals which are less than 35 dB, it will not acquire. So we are in this study, we are not interested in physiology. So we kept a little higher threshold so that we only get waves which correspond to behaviors. Okay, so now I'm going to visualize the results in from the R6A sensors using one particular feature. So there is a feature called the peak frequency of these generated waves. So I'm going to see that particular peak frequency for this 5-minute test. So for the 5-minute test, whenever the animal moved, or even when there was a small movement, there were waves, and these wave packets has been discretized so that each wave packet corresponds to a particular movement, even it is like the smallest, subtlest for a fraction of a second. The fraction of a second could be even like a microsecond or so. um, that particular waveform, wave packets, peak frequency is plotted here. So what you see is like even the smallest, tiniest movement, which cannot be even captured with the camera, is actually resulted in a wave, and then you are seeing that. And now there were so many behaviors here, like you saw, there was sniffing, and there was like rearing and grooming and all that in the video. But then you're seeing a consistently, like you're not seeing much variation here, and that is because you're only looking at uh, uh, one particular feature. Now if I combine another feature of the another feature of this waves, That is, like I am plotting peak frequency, but now I scale the size of the marker with another feature called the counts, which actually counts is something very closely related to the duration um, of the of the wave packet of the respective wave packet. So now when I represent two features, you can already see a variation in the data, and that variation corresponds to the variation in the behavior. So the point is, like, if we were to combine all the features, of course you cannot plot all the features on a two-dimensional graph. But if we were to combine all the uncorrelated features with a deep neural network model, you will be able to actually classify behaviors. And we believe these behaviors in this multidimensional space, like in this feature multidimensional feature space, each behavior is occupies a particular region. So basically, when you have a deep neural network model, it is going to separate these regions, and you will be able to track, classify behaviors just based on this acoustic data, based on this acoustic emission data. So whatever I have described so far only shows that this technology works. Like, this acoustic emission technology works, and you can track things, and it has the potential to track behavior and physiology. But I did not give you, so far evidence that behavior has a unique signature. Like, you need that evidence, right? Like, I just showed some data. Nobody knows whether uh, when behaviors are each behavior generates a unique set of a wave with a unique signature. So you need proof for that. So in this slide, I'm going to give you that proof. So in the previous two graphs plots which I just showed you, that was actually from these R6 sensors, which measured lamb waves in a small frequency region of 30 to 100 kilohertz. Now, in the experiment, I also told you I use this SH sensors to measure the shear waves in a frequency band of 70 to 230. Now, unlike the R6 sensors, the SH sensors are not fired for all the movements. You can see that it is very sparse. The hits are very sparse here. And when we actually when we clustered these hits and went back to the video and saw what happened at these moments, we found that SH sensors are triggered only when the mice either rears or when it sniffs. So what you see is the squares actually correspond to rearing, and the black dots correspond to sniffing. So only when the animal is like rearing or sniffing, these sensors are triggered. And what's more unique is you can also see that both supported and unsupported rears actually trigger these sensors. So in other words, other simple words, what means is like rearing and sniffing sometimes, most of the rearing and some sniffing actually generates shear waves. So there is a uniqueness here, like most of the rearing and some sniffing, the motions associated with them are uniquely generate the shear waves in this particular frequency range. So now this actually tells you about the unique signature in the guided waves with corresponding to behavior, certain behaviors. And also the other thing I would like to notice is like within the supported rears, for example, cluster number 10 and 8, both of them are supported rears. But like it supports the wall and then rears. But what happens is you see the frequency, peak frequency for this cluster and that cluster, 8 and 10, they are different. So what it says is like when we look at it as visually, we see, oh yeah, both 8th and 10th rearing was the same. They were just supported rearings. But what this guided wave data shows you is even within the supported rearings, there is subtle variation. But we are not capturing that thing. We can, meaning like we are not visually able to differentiate that. But there is something here, a variation within the behavior. So not only this guided wave data, now these results show you that the guided wave data has unique signatures, but it also can pinpoint these variations within a behavior. Yeah, so a couple of these were consistent results. When we tested other mice as well, other C57, we were able to see the same kind of results. Like consistently, these SH sensors are triggered when the animal rears and sometimes sniffs. So also, not all rearing results in SH modes, and not all sniffing. Some of the rearing we see, like, did not generate in this SH modes, and that could be because we are not covering the entire SH mode range. So we want, like I said, the sensor has a limited range. So that also tells you that there is, though it's all rearing, there is some difference within variation within the rearing. So uh, 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 we hypothesize, right? Once we are able to cover the entire frequency range, like the from say 20 kilohertz to 700 kilohertz, and then if you build a deep neural network model, you will be able to classify all these behaviors, and this is the necessary proof for that. So the next set of results I'm going to show you is a more even more quantifiable, even more quantified uh, observations for a particular behavior, especially the acoustic startle response. So in this study, what we did is we slightly modified the setup. So what we did is we have this 3D open field, sort of transparent open field, where we would be able to track the movement from all three axes, like from top, side, and front, visually, while also collecting the acoustic emission data. And what we do is there are speakers to the attached to the open field, and these speakers, as the mouse is exploring the open field, these speakers play like a loud tone for 10 millisecond long. And there were two types of tests. In one where we varied the frequency of the tone, like 4 kilohertz, 8 kilohertz, and all the way up to 20. And another test where we actually varied the level, sound levels, starting from 104 dB, where there's like a very mild, acoustic tone, to very loud 130 dB. And we try to see the measure this acoustic startle response from the features of these guided waves. And also the other thing we did is, like, we were able to connect this tone measure. So the tone from the laptop is played in the speakers. We were also parallelly measured this tone with our acquisition system. So we can actually estimate the amount of time it takes for the animal to react to the tone. So basically, you play the tone, and how quickly does the, like, or how much time it takes for the animal to sudden react to this tone. We were able to measure that with very high accuracy. So I'm first going to just show you the result in one of the features. So what this plot shows you is, like, there was like a 5-minute open field run, and during that 5-minute open field run, these red lines uh, indicate the instances where the tone was played, the exact instance, the time where the tone was played, and the red boxes on the plot actually is like, as the animal moves, it generates waves, and we are plotting the energy associated with these waves. Like, for every small movement there is a wave packet, and we are plotting the energy. And what you see is the red boxes, the red squares, which correspond to the startle response. Like, when you play this tone, the animals suddenly startle, and then it applies a large amount of force on the structure, on the plate, and now you see the waveforms, the energies associated with them. If you see the energy associated with most of the hits, like any other, like normal motion, they are in the order maybe of 10 power 3 or something. But when you see the energies associated with the acoustic startle, they are in the order of 10 power 5, like 33,000, 527 attojoules or something, for first startle. And similarly for the fourth startle, it's even higher. So this is one particular feature. Like, you're just looking at the energy associated with that. But similarly, you can measure other features as well, and each feature reveals a certain concept, or a certain factor of the physical reaction. For instance, we believe the energy associated with this, the energy associated with the startle response could be an indication of the sort of the anxiety, or the anxiety-like states, or the stressful states of the animal. If we were to take the other features, say, so here is the, like, the values of the other features for the acoustic startle test. Other features reveal other things. For example, the reaction time tells you how long it took to, actually, the physical force to be applied for to the startle tone. And you can see more or less it is concentrated around 10 milliseconds. And the other thing is, this is also an indicator of whether the animal startled or not. So there are dedicated studies where people try to find out whether there was a startle or not. And now this quantity factor, reaction time, is an indicator whether the animal startled or not. Similarly, other factor is the duration of the hit. That is, when the animal startled and it applied this sudden force, large force, how long did that force go on? So that is given by the duration of the hit. And the other one is the amplitude. So this is an indicator of the force, like when the startle force applied by the animal, um, force applied by the animal, how much is that force levels be force levels are. So when compared to, so the you look at the last column, so that gives what is traditionally done with videos. That gives you the pixel changes corresponding to each startle. So the total number of pixel changes, like which is done by from the video. So that is given in this last column. And so instead of it just gives you a percentage of the pixels. Instead, how much information can you gather from that? Compared to that, you see the multidimensional information data, which is the multidimensional information you get from the acoustic emissions. So okay, so this is like this pixel changes was actually calculated from our open field, from our 3D open field, where it allows you to see from all different angles. And so this is like just before the left images or uh, the images just before the tone plate, and the right one or the ones after the tone plate. And the extreme right is actually the pixels which were changed, because of this reaction, because of this response. And that is how we quantify the pixel change. And the point is, like, even with a very detailed video analysis, you only get so much information, but an acoustic emission data gives you a much more information. So the second thing about this, doing this open acoustic startle in open field is, I believe we are one of the first groups where actually we have been able to do quantify a startle response to such high precision and accuracy in a free moving mouse. Like, you can see that, and the startle response, in a mouse which is like, not constrained at all from these images. And what it allowed us to, what was the another benefit from this was, like, we were able to see variations in the startle response. For instance, in the first startle, what we observed was that once the tone played, the response was such a way that the hind paws actually retract first. The hind paws retract, and then the hind paws retract inside the body, and the front paws rise off the floor. So this was the pattern, physical pattern. But the last fifth startle response, what we saw was, when you play the tone, the front paws actually go off the floor, and then the body retracts back. So we are seeing now variations within the behavior, because like, we are doing this test, in an unconstrained environment. And actually, we were able to classify based on, we did an extensive study of the startle responses, and we were able to classify the startle responses into four categories, and we call that retraction, jump, head or body jerk, and halt or freezing categories. Yeah. So I'm just going to show the same videos, for these two starters, the first and the third starter, the first and the fifth starter, so you can actually see them in real time. This is only a few seconds now. Yeah. So this was the first startle. What I said, like, there is that hind paw retraction, and this is the fifth startle, where you could see the front paws, four paws, sort of like first going off. So this was one of the, like I said, the variation in the behavior. We could, not only quantify the behavior, but also observe variations within the behavior. And the another factor about this, doing this acoustic emission based analysis was, uh, we were able to identify whether there was a startle or not, which was, in video we could not. So the previous two videos, you can actually see the startle response. Like, a person can actually say from the video the mice startled or not. And we did another test where we varied the sound levels, and for really low really low levels of sound, there was no response. And as the sound levels increased, the mice did react to the loud tone. And but from the video, we cannot say for the really low tones whether there was a startle or not. But based on the acoustic emission data, we were able to say that there was a startle. For instance, for this 108 dB, the acoustic emission data says that the mice did start. It took a reaction time of 10 milliseconds. But uh, when you look at the video, you will not be able to say that. So now I'm going to play that particular video where you can see that you will not be able to see whether there was a startle response or not. Let me play one more time. So basically, there was a startle response. It's like a very small jerk of the head, and that is very difficult to even for an experienced scientist to differentiate. But with acoustic emission data, you can capture this really small jerk of the head. So this is another, just to showcase the what this technology is capable of doing. And to compare this to existing what we have been able to do in terms of acoustic startle compared to what is actually the traditional norm. So what you see is actually from another article. You typically do this acoustic startle in these stability meters where the animal is strictly constrained, or even like even if some of the studies which are published, they want to have a little free moving mouse. How free, even that is like sort of limited. So that is shown in this B figures. You have free, but it's not really free. It's not that free. It's free compared to A, but it's not that free compared to what we do. So this is another one of our contributions when it comes to this behavioral analysis. Um, so these things now showcase the effectiveness of this technology, what can be done with it. And with in future, what we plan to do is, we first is like, we know this now, this technology is reliable, we'll be able to do a lot of stuff with it. And what we do is like two things. First is measure physiological parameters such as heart rate and respiration in freely moving mouse, like non-invasively, in this open field what we have built. And we think we can do that by lowering the threshold. Like in these studies, we actually kept a little higher threshold of 35 dB because we wanted to study behavior. And we can measure these involuntary forces once we lower this threshold really low. And we'll be able to distinguish these hits which are coming from physiological processes from the behavior based on the uniqueness of these signatures. And the second goal of our research is like to identify this hidden behavioral models and variation within behaviors. And for that, what we plan to do is like, so far we've only looked at a very small slither of the frequency range. So we are planning to cover the entire frequency range with this really wideband sensors. And we are going to have this workflow where we will have a video output, a three-dimensional video output of the mouse, and combine that with our acoustic emission data to build a deep neural network model, which will not only be able to classify behavior based on acoustic emission data, based on these guided waves, but which will also pinpoint these variations within behavior. There was a change in rearing. There was a change in, there's like a subtle change in sniffing. And those things it will be able to identify, and then we can look at the video and see why or what behavior these variations come from, and also identify hidden modules. So at the end of these things, we think that we'll be able to comment on these measurements. We'll be able to comment some of this, effects of fear and stress on behavior, and other pharmacological manipulations on the behavior, as well as differentiate certain things which are not that easy, like say freezing from say like a non-fearful immobility and other things like that. So this is sort of the summary of my research, as well as what we are looking into for future. I would like to conclude this talk expressing my acknowledgements to both the listener. Thank you, for sitting through this talk, and also to my professors. So I am at the University of Texas Austin. And I work with Professor Michael Drew. I'm a postdoc under him in neuroscience, as well as a postdoc under Dr. Salvatore Salamoni in the engineering department, civil engineering to be particular, and also the members of both these labs, especially Dr. Laura and Dr. Nathan from the Drew lab, as well as Dr. Stylianos and Nathan from the Smart Structures Engineering lab. And a huge thanks for to the National Institute of Mental Health for sponsoring our research, for supporting our research with the grant. And we look forward to continued support, as well as continued research which will benefit the neuroscience community and the greater scientific community as well. Any queries, please feel free to contact me on my email. um, Thank you. Thanks a lot. And thanks for providing this platform to present my work.